Hello everyone, I am Kajal Jindal, Assistant Professor in Department of Biotechnology at SDPG College, Panipat, under the aegis of Director General Higher Education, Haryana. Today, I will be discussing with you the components of the immune system. So in this, we will talk about the types of immunity and what all immune responses are involved in it. Before starting, I would like to introduce you to the immune system. The immune system is a defense system that protects the body against infection or any other pathogenic microorganism such as bacteria, fungi, viruses, protozoa, etc. The immune system involves various types of cells and proteins that keeps a record of every microbe or pathogen it has ever encountered so that it can quickly recognize and destroy the microbe as soon as it enters the body again. Next, coming to immunity. The immunity is derived from the Latin term immunis, which means the state of protection from the infectious disease. It has both a less specific and a more specific component. The less specific component is known as innate immunity and the more specific component is known as adaptive immunity. The innate immunity provides the first line of defense against the infection. So, here you can see that the immune system is of two types, the innate immune system and the adaptive immune system. The innate and the adaptive immune system involve various cellular and molecular components. The cellular components involve various types of cells such as macrophages, neutrophils, dendritic cells, eosinophils, mast cells, basophils, and natural killer cells. These are the cells of the immune system, and the molecular components involve various kinds of molecules such as cytokines, chemokines, and complement proteins. While the adaptive immune system cellular components involve various kinds of B cells and the T cells, and the molecular components involve antibodies. So now in the next slides we will talk in detail about the innate immune system and the adaptive immune system. Innate immunity. The innate immunity is also known as natural immunity since it is present since birth and it consists of many factors that are relatively non-specific. That is, it can operate almost against any kind of foreign molecule or pathogen. The innate immunity provides the first line of defense against the infection and uniform in all members of a species. The innate immunity involves various types of phagocytic cells such as macrophages, neutrophils, and barriers such as skin, and a variety of antimicrobial compounds synthesized by the host. These all play a very important role in the innate immunity. Also, the innate immunity provides rapid response within minutes and has no memory. Therefore, the innate immunity does not provide long-lasting immunity to the host. Now coming to the barriers to the innate immunity. The innate immunity comprises four types of barriers: anatomic barrier, physiologic barrier, phagocytic or endocytic barrier, and the inflammatory barrier. The anatomic barrier include skin and the mucous membrane, where the skin acts as a mechanical barrier which prevent the entry of the microbes. Also, the acidic pH of sweat and the sebaceous glands having a pH in the range of 3 to 5 also prevents the entry of microbes. Similarly, the respiratory and the gastrointestinal tract are lined by mucous membrane. These mucous membrane entraps any foreign microorganism. Also, the cilia present in the lower respiratory tract propel microorganisms out of the body. And the normal flora present in the mucous membrane, they compete with the microbes for attachment sites and nutrients. Next, the physiologic barrier. Physiologic barriers such as temperature, where low body temperature or fever prevents the growth of some pathogens. The low pH in the stomach too prevents the growth of the microorganisms, and a variety of chemicals mediate protection against microbes such as lysozymes, interferons, complement, toll-like receptors and collectants. The lysozyme destroy the cell wall of bacteria. Interferons, it induces antiviral state in uninfected cells. Complement, it is a set of serum proteins which lyses microorganisms or facilitate phagocytosis. Toll-like receptors, they recognize microbial molecules and they signal the cell to secrete some immunostimulatory cytokines. And the last is collectants. They disrupt the cell wall of pathogens. The third one is phagocytic or endocytic barrier. It includes various cells that internalize and break down foreign macromolecules. Also, it includes specialized cells such as monocytes, neutrophils, tissue macrophages, dendritic cells, which internalize and digest the whole pathogen. And the last one is inflammatory barrier. The inflammatory response is mediated whenever there is tissue damage due to a wound or any other invading microorganism. The inflammatory response induces leakage of vascular fluid containing serum proteins with antibacterial activity and also the influx of phagocytic cells into the affected areas. These phagocytic cells will engulf the pathogen and helps in neutralizing their pathogenicity. Now coming to the second type of immunity, that is the adaptive immunity. It is also known as specific or acquired immunity because it is capable of recognizing and selectively eliminating specific foreign antigens. It does not come into play until there is an antigenic challenge to the organism. The adaptive immunity is not same in all members of a species. Also, it responds to the challenge with a higher degree of specificity, and it also has a remarkable property of memory. The acquired immunity display four kinds of characteristic features. The first one is antigenic specificity, second is diversity, third is immunologic memory, and the fourth one is self and non-self recognition. So if I'll discuss about it in sequence. So first one is antigenic specificity. In this, the immune system is able to discriminate among different groups of antigens. Second, diversity, which allows the immune system to recognize billions of unique structure on foreign antigens. Third one is immunologic memory. Once the immune system has recognized and responded to an antigen, it exhibits immunologic memory. So, second encounter with the same antigen induces a heightened state of immune reactivity. Therefore, immune system can provide long-lasting immunity to many infectious diseases after an initial encounter. And the last one is self and non-self recognition. In this, the immune system has the ability to recognize his own molecule, that are self molecules, and that are foreign molecules, that is non-self molecules. So the immune system will produce a response against the molecules that are non-self. Now there are two types of cells that are involved in the adaptive immune system. The first one is B lymphocytes and the second one is T lymphocytes. So I'll be first talking about the B lymphocytes. The B lymphocytes originate and mature with the bone marrow. These B cells express a unique antigen binding receptor or membrane bound receptor known as antibody molecule. As you can see in this diagram, the B cell on its surface has expressed receptors that are known as B cell receptor or membrane bound antibodies. So after interacting with the antigen, the B cells differentiate into antibody secreting plasma cells and the memory cells. Antibodies are the antigen binding glycoproteins that are synthesized exclusively by the B cells. Also, the B cells serve as an antigen presenting cell. I'll talk about this in further slides. So, whenever a naive B cell is one that has not previously encountered the antigen. So, when it encounters the antigen for the first time that matches its membrane bound antibody, the binding of antigen to the antibody causes the cell to divide rapidly and its progeny differentiated into memory B cell and the plasma cells. These plasma cells will further secrete the antibodies. The memory B cells, they have a longer lifespan than the naive B cells and they express the same membrane bound antibody as their parent B cell. The plasma cells produces the antibodies in a form that can be secreted and have little or no membrane antibody. They are the only type of cells that are capable of producing antibody molecules and therefore they are the central component of humoral immune response. I'll talk about this humoral immune response in further slides. So the next type of cellular cells in adaptive immunity are T lymphocytes. The T cells arises in the bone marrow, but they migrate to the thymus gland for their maturation. During their maturation within the thymus, the T cells come to express a unique antigen binding molecule called T cell receptor on its membrane. The T cells can recognize only antigen that is bound to cell membrane proteins called major histocompatibility complex molecules, also known as MHC molecules. These MHC molecules, they play an important role in processing and presentation of the antigen. There are two major types of MHC molecules, class 1 MHC molecules and class 2 MHC molecules. The class 1 MHC molecules, they are expressed by nearly all nucleated cells, and nucleated cells such as cancer cells, virus infected cells, etc. While class 2 MHC molecules are expressed by antigen presenting cells such as macrophages, dendritic cells, B cells, etc. So when a naive T cell is one that has not encountered any antigen before. So when a naive T cell encounters an antigen for the first time in combination with the MHC molecule, the T cells proliferate and differentiate into memory cells and the effector T cells. There are two major subtypes of T cells. The first one is T helper cells and the other one is T cytotoxic cells. They both are distinguished from one another by the presence of either CD4 molecule or CD8 membrane glycoprotein on their surface. The T cells that display CD4 function as T helper cells, while the T cells that display CD8 on their surface function as T cytotoxic cells. T helper cells. As the name suggests, these helper T cells, they help other cells of the immune system in their activation. The T helper cells, they provide protection against both extracellular as well as intracellular pathogens. The T helper cells express CD4 positive membrane glycoprotein on their surface. These T cells can only interact with antigen when presented by MHC class II molecule on the surface of an antigen presenting cell such as macrophage. So here you can see in this picture the MHC class II molecule is bound with a peptide and it is presented on the surface of a macrophage. Now this T cell present T cell receptor on its surface, and this T cell receptor will recognize antigen whenever it is bound with MHC class II. This complex formation leads to the cell activation, and then these T cells differentiate into effector T cells which secrete various kind of growth factors called cytokines. These cytokines, they are small secreted proteins or glycoproteins that are released by the cell which help in cell to cell communication. The cytokines further help in activating B cells, T cytotoxic cells, macrophages and other cells that participate in the immune response. T cytotoxic cells. These T cytotoxic cells, they express CD8 positive membrane glycoprotein on its surface. Also, they provide protection against only intracellular pathogens such as viruses and some kind of bacteria and parasites that multiply in a host cell cytoplasm. When a naive T cytotoxic cells bind to antigen MHC class I molecules, they become activated and differentiate into effector cells called cytotoxic T lymphocytes, also known as CTLs in short. The function of cytotoxic T lymphocytes is to kill cells that have been infected by bacteria and viruses. Also, these cytotoxic T lymphocytes, they are involved in killing of tumor cells and transplanted foreign cells during graft rejection. So here you can see in this picture that the T cytotoxic cells, they present CD8 positive membrane glycoprotein on their surface, and this T cell receptor will only recognize the antigen when presented by class I MHC molecules on the surface of infected cells. So when this complex formation takes place, the T cells differentiate into cytotoxic T lymphocytes and will release perforins and granzymes. These perforins and granzymes, they help in the elimination of the antigen. So the perforins form pores in the plasma membrane, while the granzymes are a set of serine proteases that enter the cell and break down proteins, thereby lysing the cell. So in this picture you can see that the infected cell is lysed. Now there are two types of response that are produced by the adaptive immunity. The first one is humoral immune response and the second one is cell-mediated immune response. The humoral response is mediated mainly by the B cells, while the cell-mediated immune response is mediated by the T cells. These both interact with each other to achieve the final goal of eliminating the antigen. The first one is humoral immunity. The humoral immunity can be transferred from an immune individual to a non-immune individual by the administration of serum antibodies. It involves B cells that interact with antigen or pathogen that are freely circulating in the blood, and upon activation these B cells, they get differentiated into memory cells and plasma cells. These plasma cells will produce antibodies. The antibodies function as effectors of humoral response by binding to antigen and thereby neutralizing it or facilitating its elimination. So here in this picture of humoral immune response, you can see that these B cells, they interact with the antigen and they get differentiated into antibody secreting plasma cells. And when they secrete antibodies, they bind to the antigen and facilitates its clearance from the body. And in this way, the humoral immune response works. Now, the second one is cell-mediated immunity. The cell-mediated immunity is transferred only by the administration of T cells from an immune to a non-immune individual. It involves mostly T cells and respond to any cell that display aberrant MHC markers, including cells invaded by pathogens, tumor cells, or transplanted cells. When T cells encounter an antigen combined with MHC, they activate and differentiate into effector T cells. Both T helper cells and cytotoxic T lymphocytes, they serve as effector cells in cell-mediated immunity. Effector T helper cells will secrete cytokines, which will then activate various phagocytic cells, enabling them to phagocytose and kill microorganisms more effectively. While the cytotoxic T lymphocytes, they participate in cell-mediated immune reaction by killing altered cells and also help in killing of virus-infected cells and the tumor cells. So here in cell-mediated immunity, the T helper and the T cytotoxic cells respond to an antigen by producing cytokines. These T helper cells, they will produce cytokines, and these cytokines will further activate B cells, T cytotoxic cells, and macrophages and various kinds of other cells of the immune system. While the T cytotoxic cells, they respond to antigen by developing into cytotoxic T lymphocytes, which mediate killing of altered self cells such as virus infected cells. So, here is the difference between humoral immunity and cell-mediated immunity. The immunity that is mediated by the B cells or the antibodies produced by the B cells found in the body fluid is known as humoral immunity, while the immunity that identifies and destroys infected cells in the body is known as cell-mediated immunity. The humoral immunity is mainly mediated by B cells, while the cell-mediated immunity is mainly mediated by the T cells. The components of humoral immunity involves macrophages, B cells and T cells, while the components of cell-mediated immunity involves T cells, cytotoxic T cells, natural killer cells and macrophages. The end result of the activation of humoral immunity is the differentiation of plasma B cells into antibodies, while the end result of activation is the secretion of cytokines in cell-mediated immunity. In humoral immunity, it can recognize antigen or pathogen that are freely circulating in the blood or the lymph, while the cell-mediated immunity responds to any cell that displays aberrant MHC markers, including cells that are invaded by pathogens or the tumor cells. In humoral immunity, CD4 positive T helper cells are involved, while in cell-mediated immunity, both CD4 positive and CD8 positive T cells are involved. The onset is rapid in humoral immunity, while the onset is delayed in cell-mediated immunity. So, this is a self-evaluation test. Here you can go through the questions and attempt all those 10 questions, and here is the answer list, and this is the reference which you can follow for your further reference. I hope you like the video. Thank you.